a discontinuous variation? Okay, wow. Continuous? Okay, all right. So some of you think that it can proceed via both kinds of variations, doesn't have to be one way or the other. That's good. Fine. Any comments, questions regarding what we did last time, where we stopped last time around, or anything else related to the course before we go ahead?
Sir, is the distribution of any attribute normal in nature?
When we're talking of attributes, there are certain types of attributes, or as in the field we would call them phenotypes. There are some attributes or phenotypes which are called quantitative in nature. And we've been using height as an example of a quantitative trait. Uh, the distribution of these quantitative traits is normal. Okay, so think height, weight, length of neck, length of limbs, and so on and so forth. So all these traits are distributed in a normal distribution. And the reason, and we'll come to the reason will become apparent by the end of the class today. But there are other traits which are not like this. And can you so can you think of a trait which is not distributed like this?
Uh, like color for uh,
Absolutely.
particular subcontinent.
Absolutely. Or color of the eye, you know. So for a long time it was thought that eye color is a purely Mendelian trait, what we discussed last time. Uh, but now we know that there are at least 7 or 8 different genes which come together to decide the color of the eye of a person. But the distribution of eye color is not going to be normal because you have different colors, right? Black, brown, blue, green. Of course, there is in different populations different colors are present. But that's a trait that's not normally distributed. But all quantitative traits, like I mentioned, weight, height, these are normally distributed for reasons again that will become apparent. But fair question. Others? Anything else?
So, and also while talking about that blending theory that Darwin couldn't justify about that, like if he proposed that blending theory, the variation should get reduced. But
Yes.
without considering blending theory also, like why can't the variation get reduced there? Uh,
No, so
hmm.
I'm not sure I understand the question entirely, but the point is that in sexual reproduction, you need a rule because two people come together and mate, and the mating between these two individuals is going to decide the phenotype of an individual. So you need some sort of a rule, and the rule that blending came up with is that h of the offspring is equal to h of parent 1 plus h of parent 2 divided by 2. That's blending, right? On the other hand, you could think of other kinds of rules, like max of h1 or h2 or min of h1 or h2, or other rules like that. And and what it does to variation remains to be seen. I mean, it's simple enough to calculate, but blending was something that was consistent the with the empirical observations that Darwin saw around him. Does that make sense?
So I mean, like without blending also, like if few attributes or few features are getting extinct or erased away, so then the distribution might change, right?
No, distribution might change, but that's one of the things that Darwin noticed, that as you go from generation to generation, the distribution doesn't change in the wild. So he's having to reconcile these things. That, like for instance, he looks at a deer population in a park, and he knows that the height distribution in a deer population doesn't change from one generation to another. So the normal distribution stays exactly the same, but he knows blending also operates. So he knows that blending should reduce the width of that distribution. So while he knows blending to be true, which should reduce the distribution, he also knows that in nature, actually the distribution remains the same more or less in a generation. There is definitely not a reduction by a factor of 2 in variation in one generation. So, and then he's forced to concede that there must be some external agency which adds to the variance and hence brings the distribution in the successive generation to the same distribution as the original generation. Uh, and this external agency of variation needs to be present because blending has reduced it by 2. So you need another distribution, another agency which would increase the variation and bring it back to the original status. Uh, but you could think of other rules also which would change the distributions, and we'll look at some of them today.
Okay, sir.
Other questions? All right, cool. So I see a couple of new people. Gaurav, Isha, can you stick around after class? I'd just like a word with you after we're done. Okay. All right, let's get started then. So we will try and explain why was this debate present, and while the debate was in certain context, we will not go very deep into the actual debate that happened in the last decades of the 19th century. But we will sort of try, our approach will be to try and reconcile the debate with what we know today. Okay. And those of you who do not know Central Dogma, that is where I want to start. So I just want to make sure that everybody is on the same page. I will go slightly fast in the initial bit, but then if this is the time, if you would like me to stop and clarify certain issues, just feel free. I'll be happy to go slower. All right. So central dogma of molecular biology, essentially the question that we're trying to answer is this, that we are all products of our genes. Our genes are catalysts that are present. The proteins that are inside us are the ones which are the workhorses of a cellular function. And they catalyze everything. They give structural integrity and all sorts of issues. So the physical manifestation of me as an individual is dependent on what proteins I can make. Proteins are chains of amino acids, and what proteins I can make is actually dependent on what is the DNA sequence that I'm carrying. So the information to make particular proteins is contained within the DNA of every cell that I possess. To give you an example, what happens in sickle cell anemia?
The RBCs are not able to hold their shape.
And that is because of a mutation in the DNA.
Point mutation.
Yeah, there is a single mutation, and RBCs, their oxygen carrying capacity is reduced. Perfect. So that is because I am carrying a particular mutation such that my RBCs are misshapen, and as a result, I don't do very well as an individual. When is it beneficial to carry that mutation?
Malaria.
In areas where malaria is highly prevalent, the malaria parasite doesn't do very well if you are carrying that variant form of RBC. So So, and then there are some population genetics consequences as a result of that. Maybe we'll look at some of them later on. All right. So the point is that we are dependent on what proteins we can make, and what proteins we can make is sort of written in the DNA that we carry. DNA is double helical. This is not a double helical, but you get the picture. So briefly, central dogma of molecular biology tells us that how is information read from DNA, resulting in fully functional proteins which can carry out their cellular function. DNA is double-stranded. You can look up the structure. You will need to understand this pretty well. I'm just going to cover some superficial aspects of it, but you will need a greater understanding as we move ahead and read some papers on this. So first thing that happens is a machine called RNA polymerase comes and undergoes a process called transcription. This happens to a certain part of DNA. And as a result of transcription, you get a molecule called mRNA. mRNA is also a polymer, which is made up of AUGC as the participating monomers. DNA is double-stranded, mRNA is single-stranded, DNA is extremely stable, mRNA is very unstable. But we don't need to worry about the chemistry of it so much. Then once the mRNA is made, you have a machine called ribosome come and sit on it, and it does translation. And it's basically reading this sequence, and while it's reading the sequence, every part of this sequence corresponds to a particular amino acid. So it reads the first 3 bases here of the AUGC, and that triplet corresponds to a particular amino acid, say amino acid number 7. There are 20 different kinds. Then it will read the next triplet, and then it will, to that original amino acid, it will add another one, depending on which triplet was present here. Let's say amino acid number 13, and so on and so forth. It will keep doing that till somewhere around near the end of the mRNA, there will be a triplet which corresponds to stop, that no more amino acids addition required, translation is complete, and hence ribosome dissociates from here and leaves, and mRNA is free again. This process is called translation, at the end of which you get a chain of amino acids. Now this amino acid can't serve its function. It needs to fold accordingly, and that is dictated by the translation, the kinetics of the translation, and the thermodynamics of the folding. It folds into its minimum free energy structure. And once it's folded correctly, now it can coordinate the cellular function that it's supposed to carry out. This process is called central dogma, but I would like you guys to look up a few more molecular details associated with this. Questions with this? Anybody who's not familiar with central dogma? Okay. Anybody else? All right. Did what I say made sense to both of you?
Yeah.
Yes.
Okay. But you guys need to look up more details. What I discussed isn't really enough. All right. So what should be apparent here, that which amino acid gets incorporated depends on the mRNA structure, and mRNA sequence, and what is the mRNA sequence is actually dependent on the DNA sequence. Hence, whatever protein that you make at the end of this process is actually dependent on the DNA sequence that you are carrying. A single nucleotide change here, say A going to a G, is going to mean that you have a different mRNA sequence, which may mean that you have a different amino acid, which could mean that the structure of the protein could change, and hence you will no longer be able to carry out the function, or you may actually carry out the function not as efficiently, or sometimes even better than what you were actually, how you were actually carrying it. So if evolution is about mutations, then basically what is happening is that when an individual is replicating DNA, so if a bacterium is replicating its DNA for the progeny, it has to copy the entire piece of DNA. And while it's copying the DNA, it might make certain errors in the copying. the machine that copies DNA to pass it on to share it with the progeny has some inherent error rate associated with it, which is called the mutation rate. And because of this changes in the DNA sequence that might happen because of the mutations, which of these parameters do you think can get affected because of acquisition of these mutations? What all could change in this process?
Transcription.
What about transcription?
So if the DNA is mutated, then it will transcribe differently into a different kind of mRNA.
Yes, the mRNA sequence will obviously change. If the DNA sequence changes, the mRNA sequence changes. What else?
The amino acid sequence.
The amino acid sequence could change. What
Yeah.
else?
So now if the amino acid sequence is different, so the job that it was supposed to do, it might not be able to do that.
Sure. I mean, this is all repercussions of the same thing, that one mutation leads to different amino acids and hence it affects functionality. What else could happen?
It could not affect as well.
It could not affect as well. Absolutely. So that's one manifestation, that you could have a mutation. And those of you who know the genetic code will know that you could have a nucleotide mutation take place, which would actually mean that even though you changed a nucleotide from A, let's say, to a C, but even then the same amino acid gets incorporated and actually there is no change in the amino acid sequence or the protein function. So this type of a mutation is called a neutral mutation, which actually makes no change to the functioning of the cell or the overall fitness of the cell. And when we are thinking fitness, again, we'll keep going back to the original definition that we discussed. When bacteria, it's how fast you divide. When we are thinking sexually reproducing organisms, we're thinking how many offsprings do you leave behind before you die. Neutral mutations. Perfect. Great. What else? What other aspects of this process could get affected?
Like it may also lead to overexpression or underexpression or no effect, as it was said earlier.
Yes, good. Yeah, exactly. So one of the see, every aspect of this process could get affected. Let's start here. Maybe there was a mutation here, which meant that the binding of RNA polymerase wasn't as efficient or was more efficient. That could affect the rate at which transcription is taking place. That's one. Second thing what could happen is that you could have a change. One of the changes we discussed was change in an amino acid, and sure, that could happen. But another implication of this change could be that the mRNA structure becomes extremely unstable. As a result, the half-life of the mRNA could change drastically, and which means that you're not going to have as many amino acid chains as you were having before, and hence which results in lower amounts of protein and lower functionality. That's another one. Similarly, you could have a change that increases half-life and you have more protein. That's also possible. You could have a change where, so the ribosomes come and bind at a particular site near the 5' end of the mRNA. You could have a change here such that the ribosome binding is affected, and again, it could be made less efficient or more efficient depending on what was the exact change that happened. You could have an amino acid change here, which one, as we discussed, could affect function. But also you could have an amino acid change which doesn't really affect function, but actually changes the half-life of a protein. Okay? So every bit of this process can actually be controlled and modulated by the process of acquisition of mutations and then natural selection acting on that particular mutation. Any questions or any other aspect in which this process could be affected that you want to highlight?
So because of the amino acid change, you're saying that it could also just only change the half-life of the protein.
Yes.
So does that mean it makes the protein more unstable?
Yes.
So doesn't that actually imply that the functioning of the protein is affected?
Not really. By functioning, typically you are, so proteins are essentially catalysts. So by functioning, what you are saying is what is the kcat of the kcat of the protein molecule, and by half-life, you are saying that how long is that molecule going to be around. So
What is KCAT?
you can think of kcat as that how many substrate molecules does a enzyme catalyze in a unit time.
Oh, okay.
Yeah. So by functioning, what we typically mean is that the enzyme could catalyze 100 molecules every second. But now you made it better, it can catalyze 120 per second. But by
Okay.
stability, we mean that that enzyme molecule was going to stick around for 10 minutes, but now it's only going to stick around for 5.
Okay.
Others?
Yeah. So when you said that it reduces the half-life, so in this case, in a cellular context, it would mean that it's degraded within the proteasome complex, which degrades proteins. So it could be that. Does that mean that if you have extra ubiquitination processes, which are
Yes.
actually, which would help in the proteasome in the degradation process, so that would also be consistent with what it changes, right?
Yes, yes, absolutely. Yeah.
Okay.
So we will in a few weeks read a very interesting paper that was published in the 1980s, and the argument of the paper is well. Let me first say this, that so if I were to make an argument that every single parameter associated with this is acted upon by natural selection and brought to its optimum, okay, the rate at which this is being transcribed, what should be the half-life here, what should be the half-life here, the ribosome binding site, every aspect of it is acted upon by natural selection over a geological time to bring it to the best possible solution in given a particular environmental context. This particular way of looking at things in evolution is called a very selectionist point of view. Okay, that natural selection has acted on even the most minutest aspects of gene regulation here and brought it to an optimum, and not only just transcription but every subsequent step leading to the protein amounts that result. This type of a view is called a very selectionist point of view. There is some opposition to that as well. I mean, there are other ways of looking at looking at the diversity that's present around us. And we'll discuss these when we, in a few weeks' time, read a paper written in the 80s. All right. But again, let me show of hands. How many of you subscribe? For how many of you it sort of agrees with your gut that natural selection could have optimized this entire process and every sub process within it? What do the others think? Okay.
Sorry, slightly late.
Yeah. Sure. All right. That's an interesting debate that we'll come back to later on. All right. The point is there is the prospect of natural selection acting on it. So well, I am curious. What do the others think? Anirudh, why don't you tell me why do you not feel comfortable with that point of view?
Honestly, I don't know the answer to that. Yes, I'm not
Okay.
sure.
So there is no discomfort. It's just that you don't know how to think about that.
Yeah, yeah.
Okay, cool. That's fine. All right. let's move ahead. So these we'll call evolutionary tunable parameters in this process: rate of transcription, stability, rate of translation, stability, and the enzyme kinetics, the functionality of the, and each of these can be accomplished by changes in the DNA sequence. And as we'll go forward, we'll read some papers, and we'll see that manifestations of each kinds of these 5 things that we have seen, that I've listed here, has been observed in several experimental evolutionary systems. So this is not a stretch of the imagination in any way whatsoever. All right. So remember, all of this discussion is happening in the context of our attempt to explain whether natural selection acts on discontinuous or continuous change. And in order to get to that debate, again, as I said, we won't visit that debate in a historical context because I think it's very convoluted without the molecular biology knowledge that wasn't around at the time. But we revisit it in a modern context. And all of this is just sort of to prime you up that by the time we get to end of class today, we can revisit that discussion. So what we discussed was just one gene on the DNA. However, there are, of course, several genes located on DNA. So gene A, gene B, gene C, this will be transcribed and translated into its protein. This will be, and so on and so forth. And the concept of gene regulation means basically that each one of these proteins is only made when it's needed in the environment and in the context that it serves a function. For instance, if gene A helps you utilize lactose for growth and energy, then this protein molecule will only be made when lactose is present in the environment. If there is no lactose present in the environment, making this protein serves absolutely no purpose, and hence protein A will not be made inside the cell. That is the concept of gene regulation, that you can tune the expression. You might have this information contained in your DNA, but it only makes sense for you to undergo transcription and translation if the resulting protein is going to serve a certain function inside the cell. All right. So just to give you a sense, again, those of you who have not seen, genes have names, and they are usually, sadly, not as simple as ABC. The names are actually really horrendous. So when I was doing my PhD, I worked on a bacterium called Salmonella, which causes infection via food. It's usually ingested with infected food. And one of the genes that's required is called HIL-A. So gene names would typically have three letters and then a 4th letter, and they're pronounced as HIL-A, and it stands for hyperinvasive locus A. E. coli has, or many bacteria, all bacteria have this gene which is called, some people call it FTSZ, some people call it FITZ, and this is implicated. This has a role. This is a big role to play in cell division, and FTSZ stands for filamenting temperature sensitive mutant Z. So, I mean, gene names are pretty horrendous, but for the most part, this suffices. We don't have to worry about what it actually stands for. One of the tragedies that's happened with naming genes is that when genes were discovered, the discoverer necessarily did not know what the resulting protein from this gene actually did inside the cell. So they would just name it anything. So you found, let me just make stuff up. You found HIL-A in Salmonella. You called it HIL-A because it was hyperinvasive locus. If you got rid of that gene, if you removed that locus from the DNA of the organism, you found that Salmonella can no longer infect. So you called it hyperinvasive locus. That presence of that particular locus, that particular location on the DNA is extremely critical for the ability of the bacterium to infect. Hence, you called it hyperinvasive locus A. Somebody else might have been working on another pathogen, let's say Pseudomonas, and they would have found another gene, which they, let's say, called Pseudomonas infection gene. Again, I'm making this bit up. But then DNA sequence wasn't available, and all you knew was that this region of the DNA is important for infection. So you call it something completely different, Pseudomonas infection gene X. And later on, when DNA sequences were easy to get analyzed, and when you compared the DNA sequence of these two genes, what you would find is that they're remarkably similar to each other. And the natural conclusion when you find DNA sequences which are extremely similar to each other is that they must have had a common ancestor and diverged from there. One copy went in Salmonella and evolved into today's sequence. Another copy went into Pseudomonas and evolved into today's sequence. And these two sequences hence look very much alike. Now, if we knew the sequence information at the time when the gene was discovered, then the names would be very similar, so that we would know that these are sort of the similar kind of proteins. But as I said, when the naming was done, we didn't really have sequence information or even the functionality information, and hence gene names in biology are pretty horrendous. All right, any questions with that? Any questions with the fact that there are genes located on DNA which are like this?
So I had a question. So you're saying that a gene can be expressed according to the environment. So
Yes.
different people might have the same genes but different proteins because they live in different environments.
Yes.
So is it like so I am always confused with the fact that suppose if a tribe of people are living in the mountains since like hundreds and thousands of years, their generations have been living there. So have they evolved differently, or is it just that we also have the same genes, but they have expressed the genes that require less oxygen intake, and they can survive there better?
So actually, hemoglobin is one of the proteins that's been studied extensively regarding this. People who lived in high altitudes tend to have mutations which bind oxygen more tightly. Okay, so definitely there is adaptation based on the environment that you are living in. We live near sea levels. I mean, in Bombay at sea level, so oxygen concentrations are much higher. But if you go to upper Himalayas, you would find mutations which bind hemoglobin, hemoglobin mutations which bind oxygen much more tightly than our hemoglobins. So yeah, those variations exist.
Okay,
And
so they have a bit of different genome, like they
yes,
have mutations. Okay.
yes. But you don't even, but mutation is one thing. You talked about differential expression, genes being turned on and off. You don't even need to go to different people. You need to just look at yourself. The cell that's present on your skin, on your palm, has the exact same DNA of a cell which makes up your liver, for instance, right? But they look very, very different from each other. And this cell has the exact same piece of DNA which is present in a cell in your eye. All three have the same DNA, but physically they look extremely different. And that is because all three live in different environments. And hence the genes which are actually being transcribed and translated are different in each case. Okay.
Yeah.
So you don't need to go and compare people. We have this going on right within ourselves.
Yeah. Okay.
Okay. Anything else? All right.
So while telling about central dogma and mutation, there you mentioned cell fitness. So that was related to bacteria, like
Yes.
cells are only those cells, right? Not
Yes.
the cells of multicellular organisms.
Not multicellular, but you can think. I meant it in context of single celled organisms. But you can think of it in the context of multicellular organisms also. For instance, if you pick up a hemoglobin mutation which gives you a sickle cell RBC, right? Then if you, so again, this is a very classically well studied system. if you are living in a malaria prone area, then all of us have, let's not get into that discussion right now, okay? But yeah, I meant it in terms of bacteria, and you can think of it in those terms for now. Yeah. Others?
Sir,
Yes.
you mentioned about
You go ahead. Sorry.
that's okay. So you mentioned about two infectious genes coming from the same ancestor, right?
Mm-hmm.
Why is it not possible for two different organisms to get these infectious genes by virtue of evolution without having a common ancestor?
No, just pure statistics and probability. The chances that a DNA sequence of 1,000 bases evolves to roughly the same sequence via two independent events uh, is practically zero.
Okay, so is it not even possible for a certain gene to be like that, if not the
No,
entire DNA?
no, definitely not entire DNA, not even a gene.
Okay.
Okay. But what can happen is that it need not come from a common ancestor. It could just be, and bacteria do this very well, and I wasn't planning on talking about this. What can happen is that I am a bacteria and I have a certain gene, and here is another bacteria which doesn't have that gene, and there is a process which is called horizontal gene transfer, which could happen via different mechanisms. We won't get into the details of that now. And what could happen is that these two species can trade genes with each other.
Okay.
Okay. So now what can happen is that I can give you a gene. And now this piece of DNA gets into your DNA, and presumably you found that gene of some use, and then you retain that gene. And now we both have a copy of that gene, and we will pick up minor mutations which are dependent on the precise environmental context in which we are surviving, right?
Okay.
So then we have copies of two genes which look very much alike each other, and they didn't come from a common ancestor, so to speak. This was just passed on from one species to another via several processes. Okay.
Okay.
Yeah. All right. So we'll keep
So,
moving.
and also coming to the function of the gene, like,
Mhm.
is there any case that particular gene A will get completely transformed to some other gene in the course of evolution?
Say that again. I didn't
In
understand that.
a particular gene A with some function
Hmm.
will get completely transformed to some other gene with some other function.
It is possible. Uh, it is possible, and it can happen several different ways. When you say transformed, I am guessing you mean that is it possible that a gene A originally codes for a protein which does function X, but with time you're asking if gene A can pick up mutations such that it no longer does function X but does function Y now.
Yeah.
Right. Okay. Uh, it can happen. Let me tell you the most common way it happens. Okay? The most common way it happens is that imagine that this is the piece of this is a gene on a long piece of DNA. What happens is that sometimes when the DNA is being copied, this gene accidentally gets copied twice. Okay. So what that means is now you have two identical pieces of DNA with the same, you have two copies of the same gene. Does that make sense?
Yes.
Right. What will happen now is that let's say that this original gene served a function which was, I mean, the protein that was encoded by this gene served a function that was critical for the survival and growth of this this particular individual. By individual, let's think bacteria. It's just simpler to think that way. So this encoded for a function which was critical for survival and growth. But now you have two copies. You don't really need two. So what can happen is that now the additional copy or the origin, one of the two copies, can pick up mutations and evolve into some other function. Because now you originally have kept you have kept one of the two copies which will keep on functioning its original function X, but now the other copy can keep picking up mutations and evolve into function Y, which could be helpful in another context. Does that make sense, Namrata?
Okay, so but that doesn't happen without copying.
So that's one way. This is called evolution by a gene duplication. Okay. The mechanism that you're talking about, if you think about it, it's only possible if function X is no longer critical, because otherwise, if loss of function X reduces fitness, then this guy will never give up the function X, right?
Yeah.
Right. So what the scenario that you're talking about is only possible if fitness X is no longer needed and function X is no longer needed and function Y enhances fitness. Right? So this is seen in the cases of genes, which encode for proteins which are called bifunctional proteins. Okay. So there are several proteins which not only carry out one enzyme, but carry out actually more than one functions inside the cell. In that case, what can happen is that the relative sort of rate of catalysis of this particular protein, this gene which codes for the protein, for function X and Y can be modulated depending on the precise environment that this individual is growing on. Right? So think of this as a bifunctional gene which can carry out X and Y. But now the environment changes that X is not so critical, but Y is very critical. So this will evolve to become better at Y and worse off at X. Right? But then environment changes and now it needs to be better at X also. So then it will sort of try and retain Y, but then try to pick up mutations which make it better at Y. So these sort of dynamics can happen and change functionality. But examples of the kind that I just talked about are not easy to find. Bifunctional proteins are not easy to look at. Does that in some way answer your question?
Yes.
Yeah. But duplication has been studied extremely extensively, and basically duplication happens, and then you have a free copy which could evolve into absolutely anything. And that's thought to be a major source via which novelty evolves, because now you have a copy of a gene. What we've been talking about, point mutations, A changing to G, A changing to T, and whatever, can change rate of transcription, can change function, can reduce degradation rate, increase degradation rate, stability, can bring function catalysis up or down a little bit, but it can't introduce new functionality, just something completely new. Something completely new happens via this mechanism where a gene is copied or a set of genes is copied, which can now evolve into something else, or new functionality is acquired via the previous question that I just answered, that this is a bacteria which has a set of genes and it just passes this set of genes to another bacteria. And now you have a whole battery of genes that you have just acquired, which can evolve to serve a function which you previously couldn't do. Does that make sense, everybody?
Yes.
All right. Okay, so let's move on. So, I mean, just some sense of numbers for those of you who don't know. So a typical bacteria will have maybe 5,000 different genes. The genome length might be of the order of 10 parsecs. And in order to compare that with us, we have an order of magnitude 10 power 4 genes, and our DNA length is 1,000 times bigger than a bacteria. What's interesting is that while this is 1,000 times, this is 3 orders of magnitude larger than a bacteria, the number of genes isn't nearly as much greater than the number of genes that a bacterium carries. But there are reasons that we don't want to get into. All right. Let me ask you a few questions now. How we will settle this debate? We have 45 minutes, and I want to be able to finish this debate now, continuous, discontinuous variation. We want to look at how evolution happens in a fundamentally different way in a population of bacteria or yeast, such as what we would do in our lab, and in a population of pea plants, as Mendel was doing, or human beings as we progressed as a race. Evolution happens via fundamentally different ways in these two cases, and it's not as if the two mechanisms are completely alien to each other. What happens in bacteria also happens in human beings, and vice versa. But the relative importance of these factors is totally different between these two cases. We'll do bacteria first. So let's imagine that I take 1 liter of liquid media. Liquid media basically is something which has all the nutrients for a bacteria to grow. And I add 1 bacterium to this flask. And this is, let's say this is E. coli. And E. coli loves 37 degrees. And I allow growth for several hours, let's say 24 hours. And at the end of it, the flask is just basically extremely turbid, full of bacteria. And the numbers can be up to 10 power 9 bacteria per mil of a glass. So if this is a liter, then we have 10 power 12 bacteria in this flask. If we collect all the cells, we spin them down, make a small pellet or something, and spread them on a solid plate which contains nutrients for them to grow, plus an antibiotic. So I collected all 10 power 12. I spread them uniformly here, right? But the interesting thing, so this is a solid media. It's like a jelly-like structure. Basically the same liquid, but it also has something which makes it solidified, like a jelly-like structure, so that a bacteria cannot move. Here, a bacteria can obviously swim through the liquid media. Once you are placed here, you stay here on this particular media. So I collect all the cells, I spread them on a plate, but the plate also contains an antibiotic, right? And I keep this plate for growth at 37 degrees Celsius for another 12 hours. What will I see on the plate the next day? What are the possibilities? Just tell me everything that comes to mind. Is the question clear? Is the question clear?
So antibiotic is regional, right? It's not everywhere.
It is everywhere.
Okay.
Is the question clear?
Is the antibiotic specific to that particular bacteria?
Yes. Let's say it's ampicillin.
Okay. Then it won't grow.
And the original population was sensitive to the antibiotic. So we can have two kinds of populations, a sensitive population or a resistant population. The resistant population means that the antibiotic won't be able to kill the bacterium. The ones that we started with is actually sensitive to the antibiotic. That means in the presence of that antibiotic, it will die. What are the possibilities?
Well, it's possible that you see maybe 1 or 2 colonies while the rest of them are dead, but they don't thrive.
Why would they?
Those 1 or 2 colonies had mutations of some sort, which could have helped them, but not necessarily. Most probably, I think that if you take it from a culture which is from the same strain, so I doubt that you would find. You might just find 1 colony probably out of the 10 power 9, which actually has a resistance to that particular antibiotic.
Okay.
We can also see, it might happen that the new bacteria, like it is constantly growing, but the rate of growth will be declined, and the already existing bacteria will also start to die. But some of them who are able to reproduce in the small amount of time, the new bacteria that is formed would be a bit more resistant than the others to the antibiotic.
Why would the new bacteria be resistant to them? Because there was no antibiotic in the flask that they were growing in.
So you're saying that growth will not happen at all in the
I'm not
jelly-like
saying, no, I'm asking what can happen.
It's
So
growth.
Anirudh, why do you expect a handful of colonies or 1 or 2 colonies?
Due to mutations, I guess. I mean, if it was naturally possible that maybe 1 or 2 colonies during the time in the bacterial culture, they had found mutations which helped them which helped them in
But
this.
in the environment that these mutations happened, they weren't really useful, right?
No, yeah, exactly. So it
Yeah.
wasn't useful there, but it suddenly was useful here.
Right. Yeah. Perfect. Great. So that is indeed one of the possibilities, that what might happen that we started off with 1 bacteria and we said this was a liter. So we have 10 part 12 bacteria in this flask. How many cell divisions happened in this process going from here to here?
10 to the power 6.
Others? How many cell divisions happen in this scenario where we went from 1 bacterium to 10 part 12 bacteria?
Part 12.
Yeah, 10 part 12 minus 1 divisions, right? So anyway, we'll come back to that. So what we would expect is that one of the possibilities. So I've written here what are the possibilities that exist. One of the possibilities is that you don't see anything, because none of these 10 part 12 bacteria carried the mutation. Let's say there was 1 mutation which conferred resistance to the antibiotic. And it just happened that none of the 10 part 12 bacteria, when spread on this, had that mutation which conferred resistance, in which case everybody dies. On the other hand, it's also possible that maybe 1 bacteria growing here had that mutation which when it was placed in an environment which contained the antibiotic conferred resistance, and as a result of that particular individual would grow, and then when I come in after overnight growth, I would see a lump of cells around that single bacteria, what I call a colony, right? So I might get 1, I might get 2, but I definitely don't expect thousands and thousands of bacteria. Interestingly, this experiment was the basis of a very famous paper by Luria and Delbruck in late 40s, which led to something called fluctuation test. And those of you who are in the course 715, we discussed that in a couple of weeks. It's a really fascinating thought.
So
anyway, yes.
In each iteration of the experiment, we won't see the colony, right? There might be some iterations where no colony was present.
Absolutely.
Okay.
Exactly. So because it's a chance event, right? If let's say when the first division happened, when we added 1 and that guy divided, if that guy's progeny, when it was 2 bacteria in the flask, if that mutation was picked up at that instant only, then how many colonies do you expect?
More.
But I mean, around we will see a very good distribution around Petri dish.
But then, yeah, but how many? Give me a number. So you have two bacteria in the flask now, one which is sensitive and the other one which carries a mutation which confers resistance. So at the end of the experiment, when you plate, how many colonies do you expect?
10,006.
50% maybe around.
Yeah, exactly. Not 10 power 6, 0.5 into 10 power 12.
doesn't it depend कि वो फ्लास्क में ग्रो कर पाया कि नहीं या फिर वो म्यूटेशन की वजह से वो फ्लास्क में मर गया?
Yeah, so what Akshat is saying is that one of the possibilities could be that when this mutation was acquired, when this mutation that confers resistance in ampicillin is acquired, that individual, in the absence of antibiotic in that flask, might not grow as well as the original sensitive bacteria, right? So in that case, the number of the resistant bacteria at the end of the growth cycle will not be 0.5 into 10 power 12, which will be less than that. And that is correct. That is correct. Typically, what's seen is that resistant bacteria, because they are making this unnecessary protein. I mean, if the antibiotic is not around, and they are making this unnecessary protein, that adds a certain cost to the cellular machinery that they have to make this additional protein molecules, and that additional cost reduces fitness. So if you compete a resistant and a sensitive strain in the absence of antibiotic, what's typically found is that the sensitive strain will have a greater fitness than the resistant strain, obviously in the absence of antibiotic. But I mean, that fitness advantage is not going to be more than a few percentages. So for the purpose of argument, 0.5 into 10 per 12. But yeah. Questions? Okay. I just wanted to show you this paper. I mean, it's not in our reading list, but I did want to show you this paper that came out, not paper, but this came out 3 or 4 years back. And take a look at this paper. This is from Roy Kishne's group in Harvard. Paper, I think, came out in 2016. What they did is, you can't hear the audio, can you?
No, sir.
Yeah, let me unplug it. Can you now hear the audio?
So, barely audible.
No, sir.
You guys can hear him now, right?
No, sir.
Okay. So let me explain. What they've done is made a giant plate. Okay. On the outer edges there is no antibiotic, then just the slightest bit, then 10 times more, 10 times more, and so on and so forth. And they introduce bacteria at this edge and they allow them to grow. And what will happen is, and this jelly is so soft that bacteria can actually diffuse, swim through this jelly and travel in search of more resources. So as growth will happen, what you imagine is that you will reach here. E. coli will eat up resources available in this part. The right half is just a mirror image, so we're not going to talk about that. It's going to eat up the resources here, and then it gets to this interface. It can't go beyond because it can't survive in this presence of this much amount of antibiotic. They're carefully controlled the concentration such that the strain that they're starting with won't grow in this concentration. Then what will happen is that you have to wait for one individual at this edge to pick up a mutation which confers resistance to this particular concentration. And then once it happens, it sort of starts to spread from there and eat resources here. But then it gets to this interface. Now the mutation that was confirmed here might not be enough for the concentration of antibiotic that's present in this band, and hence growth again stops here. And then it has to wait for another mutation to come, and so on and so forth. So let's just take a look at this. All right, does that make sense? Any questions about this?
So in the middlemost section, the colonies did not grow that much. Was there a significant reason?
I think the concentration was just too high. I mean, antibiotic resistance happens via two ways. One is via acquisition of mutations. And if this is a protein, this is a critical protein for cellular functioning, then then antibiotic comes and binds here and stops it from performing function. But then this protein can pick up a mutation so that antibiotic is no longer to so effectively bind. But I think the middle section is just so loaded with antibiotic that it's just really hard for this protein to pick up mutations and escape the antibiotic completely. The other mode of antibiotic, which I think would accomplish growth in the middle band also, is that if this bacteria were allowed to pick up other genes from the environment, from other species and so forth, such as the mechanism that I answered for Pavitra's question when I said that you can trade genes with other species also. That is something that will help acquire resistance to much higher concentrations of antibiotics. But in the context of this experiment, they're only starting with one type of bacteria, and that's it. There is no other kind. Soil, for example, has lots of DNA. Soil bacteria pick up DNA from their surroundings. None of that is being permitted here, and I think that's why it cannot go to that high. It cannot grow so robustly at that high concentrations of the antibiotic. But I think it's a pretty cool. I mean, the experiment is fundamentally different from the hypothetical experiment that the Lurie and Delbruck experiment that we discussed, because in the experiment that we discussed, our flask, we are growing without antibiotics. So anything that comes up as resistant is only because of the chance that a mutation happened, while that mutation did not confer any advantage. In fact, as Akshat pointed out, that mutation actually makes the bacteria worse off, but still it arose in flask and it's not going to die. It's going to grow perhaps a little slower than everybody else. But that's the reason it arose, because mutations are a chance event. In Kishney's experiment, mutations arose because there was a big selection pressure. If you didn't have that mutation, you couldn't grow. Hence, there was a big selection advantage in any individual which was able to pick up that mutation. Sakshi, yes, mutations are pretty rapid. In fact, I'm just coming to that on the next page. Okay, we need to, I'll just go a little bit quicker. So what does this mean about evolution in bacteria or asexually reproducing organisms? That you have a parent, it splits into two. One of them has the identical copy, other one picks up a mutation. This is the error in copying DNA, right? Example, A was changed to a T. The rate at which this error is made is called the mutation rate of the species. Every species has a unique mutation rate. Mutation rate is also dependent on the environment. That's some recent work. But essentially the rate at which the machine that's copying DNA is, not surprisingly, called DNA polymerase. And the rate at which DNA polymerase makes errors is called mutation rate. For E. coli, the rate at which error is made is 10 power minus 3 per cell per generation, which means that when a bacterium is dividing into two, there is 0.001 chance that the progeny has one error in it. That means you need to have 1,000 divisions take place for you to find, for statistically there to be one error in one of the individuals that was in the population. Okay? And because the genome size is of the order of 10 power 6, if you're talking of an error at a particular location, then that error rate is basically every time a base is copied, a nucleotide is copied, there is a 10 power minus 9 chance that that base is incorrectly incorporated in when the DNA is being copied. Okay? So at every nucleotide, there is 10 power 9 minus 9 probability of an error. What would be the effect of these mutations that have happened on the fitness of our bacteria? Fitness could increase. If that happens, it's called a beneficial mutation. Fitness could decrease. If that happens, they're called deleterious mutations. And these are the variations that Darwin was talking about. And fitness could remain the same, in which case, as Sangamitra said, that you could have mutations which don't do anything to fitness, in which case they're called neutral mutations. And I say other mechanisms of variation exist too, and these are the mechanisms where trade of genes happens and stuff. We will perhaps revisit this later, but for now, I just want to concentrate on errors being made by the copying machinery. So the idea is that when asexual organisms are sort of replicating, this is the mechanism by which variation is generated in the population. Variation is generated and then selection acts on this variation. If antibiotic is present in this environment, this guy will survive and this guy will have a fitness equal to 0 because this will die. If on the other hand, if antibiotic is not present, this guy, which is resistant to the antibiotic, but there is no antibiotic present here, this guy will grow slower, will have a lower fitness, and this guy will keep on growing faster as compared to this one, and so on and so forth. So that's the mechanism by which variation is generated in asexual organisms. Now, this is fundamentally different from how it happens in sexually reproducing organisms. So now we're going to think of sexually reproducing. So there's a 1,000 Genomes project. This is a few years old now. And on an average, the project concluded that a typical genome, mine with any one of yours, a typical genome differs from the reference human genome that's been compiled at 4 to 5 million sites. So 4 to 5 into 10 power 6 differences in the nucleotide sequences are there if I were to compare my DNA with the reference genome that's annotated. Now, that's a lot, but the total size, remember, is 3 into 10 power 9. So it's like 0.4% of the total DNA. That's the sort of variation that we're talking about. Something that you guys need to sort of know is that imagine I am individual A and I have a gene X, and this gene X will have a certain sequence. You are individual B and you also obviously have gene X, but your gene X has a change, right? I mean, we don't know which one is better, but the two sequences are just not identical is what I'm trying to say. Okay, so suppose we sequence gene X in each one of us. I see 15 people in this meeting. So if we sequence 15 gene X's in the entire population of this class, then we will get different variants of gene X. These different variants of gene X are called alleles. Okay. And if different alleles exist in a population at the same time, that phenomena is called as polymorphism. Okay? So let's imagine that we sequence gene X among each one of us, and we find that there are 3 different variants of gene X that are present between the 15 of us. Then we will say that for gene X, there are 3 alleles in the population being this class. There are 3 alleles in the population, and gene X is polymorphic. That there are several alleles that exist in the population at the same time. On the other hand, there could be a gene Y here, which if we sequence that gene Y in each one of us, we might find that gene Y is completely identical in each one of us. So the sequence is exactly the same for all 15 of us. In that case, we will say that for gene Y, there is only one allele in the population, and gene Y is therefore not polymorphic. Okay. Does that make sense? Does what is an allele and what is polymorphism, does that make sense?
Yeah.
Yes, sir.
All right. Cool. This is kind of important, so great. So now let's imagine, before we do this, we have 15 minutes. I do want to give you a task, few minutes. I did my PhD at, okay, let me not show you the graph. I did my PhD at University of Illinois, Urbana-Champaign, which is a land grant university, meaning that this was started in the middle of the 19th century when government gave land to the state to start a university to do research in agriculture. Illinois is primarily agricultural. And corn is a big product in Illinois. And the university is home to the longest running evolution experiment in the world. And the experiment was as follows, that there is a field and you grow corn in that field. Okay. And you're going to have different plants, and each one of them will have corn at the end of the season. What you will do is you will take corn grains, or I don't know if they're grains or whatever. If you will take corn from each one of those plants and calculate and find out, go to the lab and get the oil content in each one of those plants. Then what you will do is you will reject the 90% of the plants which had the lowest oil content. You will only select the top 10% plants which had the highest oil content. Then for next year, you will prepare the seed to be sown for the next season only from these top 10% plants with highest oil content. Okay, you will prepare those seeds. Then you sow those seeds the next season. And of course, you will have a field full of plants again. And you keep repeating the same process over and over again. Is the experiment clear?
Yes.
Yes, sir.
Any questions, doubt about the experiment? All right, cool. This experiment was started in 1896. The experiment is still going on. I want you to sketch, it'll take a couple of minutes. I want you to sketch, x-axis is time. So time starts at 1896 and we can go up to 2020. Of course, I'm not interested in numerical values, a qualitative sketch. And y-axis is the mean oil content in the plant in a particular season. So let's say we started at a certain value of 5% oil content in 1896. Okay. So y-axis is what is the oil percentage. Take 2 minutes and think of what would this trajectory have looked like from 1896 to now. Is the question clear?
Yes.
All right. Just take 2 minutes and I will be back then. All right, tell me, what do we think?
So I think it will be kind of exponential growth and then a plateau.
Okay.
Yeah, it will saturate at some point.
Okay.
Like there's a plateau.
All right. Others? I would like people to guess.
Still increasing.
Others?
Sir, I personally feel that because we are growing at such a large scale and there might be some environmental factors, so at times the growth might be fluctuating depending on the environmental factors.
Okay. But in every season, you select the top 10%. You reject the bottom 90%. Those seeds never make it past. But you're right, there is role for environmental noise. Others?
So since only like the top 10% is selected, would it be something like linear, like it's the mean keeps increasing?
So let me ask you this. Why should oil content increase? What will be the molecular basis for increase in oil content?
More expression of that particular gene or something.
What would facilitate more expression of that particular gene?
The selection pressure, maybe.
But selection pressure is not going to do it, no? I mean, there needs to be. It's not as if plant is looking at me and saying, "Oh, look, Supreet is coming. He's going to kill me if I don't produce top 10%." Environment is
Some
environment is the same for
environment.
everybody.
But drawing idea from Darwin's point of view of natural selection, that we are not allowing to the low oil percentage plants to grow.
Yes.
So the high oil percentage plants will grow in number, and eventually the oil concentration in each plant will be growing.
Let me show you data. And Prachita, I would like to keep the trail that you. This is, forget about the others. It doesn't matter. Only the green one. This was in 1896. This is 2010 or so. It's linearly increasing with time. Right? Does everybody see? I mean, there is an occasional, kink here or there, but it's linear increase. What does this tell you? Prachita.
So there's like most of the plants have, I mean, okay. There's more oil content in the plants that are grown now.
Well, that's obvious, yes. And but let's look at it like this. If we had a population, if we had a starting population which was producing certain amount of oil, and to increase oil in the plants, you needed mutations, right?
Okay. Yeah.
If that was the case, then what you would have thought is that the trajectory would look like this, that till that mutation happened, the oil content sort of more or less fluctuated around a level, depending on what was the genotype of what was the DNA of the existing plants. And then when somebody acquired a mutation, then oil content would increase suddenly, because now you have a plant which has this mutation, right? And then you're waiting for the next mutation, and so on and so forth. So you expected perhaps more a step-like response. Does that make sense?
No, I didn't understand.
Yeah. All right, this is why would the oil content, I guess we'll continue this the next time, but why should the oil content increase with time? And I'm selecting for, let's say oil content was normal distribution. Okay, it was normally distributed in the original population. I reject the left 90%. I take the next 10% forward. Right? Why should the mean change?
Because there are
So,
more plants which are producing like.
Because the mean of the 10% would be higher than the original mean of the
Okay.
previous population, and then so and so it will become higher, but it will never go beyond if you are not considering mutations, and just if the genes remain the same, then it will never go beyond the first highest value of oil content in the first batch of plants we had.
But highest से तो बहुत आगे निकल गया है.
So then mutations might have happened.
I mean, if you see this, I mean, I don't have the data right now, but if you look at the actual data, then the distribution around 5% would be something like this in 1896, right?
Yeah.
From starting from maybe 3% to 8%. The distribution now is around 20%.
Yeah.
Right? So there is no overlap.
Okay.
So, I mean, Prachita, I do want to make sure that you got the problem that we have run into. If we select the top 10%, okay, and we expect them to produce more than the mean of the previous generation, then what we are saying is that there was a genetic basis to oil content. Right? Which is fair. No problems with that, which is fair. But the problem that we have to explain here is that how could that same plant, how could that lead to 5% to 20% in this much amount of time?
Okay.
Anirudh, do you have thoughts?
Were there no plants that had 20% initially in the first?
No.
Yeah.
No, not even close. As I said, the initial distribution was like 3 to 7% or something like that.
Is it possible that one of the future, so at each successive generations you get certain mutations, which, yeah, no, it doesn't make sense.
It does?
No, it doesn't.
But I do want to clarify that what you said could not be what is happening. So one of the possibilities is that in every generation there are new mutations which account for the higher oil content. The problem is linear nature. But we know the mutation rate in corn plant. We know the mutation rate is not so high that you expect in every season a mutation to occur which increases oil content. So it's not newer mutations. I mean, over the course of 120 years, sure, there could have been mutations which increase oil content, but not every year. The field is not very large. I mean, I've seen those fields. It's not a very large field. So then how could oil content increase if it's not new mutations every year?
Is there a possibility of horizontal gene transfer?
No possibility of horizontal gene transfer.
Phenotypic heterogeneity.
But it's not inheritable then.
Why would it not be? It can be inheritable for a few generations.
It could be, but that's not the case. I don't know corn plant biology so much in order to say for sure that it's not the case, but let's say it's not. I'm pretty sure it's not, but I don't know that for a fact. Is the conundrum clear to everybody?
That it shouldn't increase linearly every season.
Yes. If it was new mutations, then you would see a step increase or a very rapid increase in the season that it happened, and then in the few seasons that it followed. And then there should be a lull where nothing happens, and you're just waiting for a new mutation to arise in the population. The population size is not very large. If you have a huge population, then you are also increasing the chances of a mutation happening. But you only have a very small population, chances of mutations happening are also small. So we have to explain the linear trend.
Sir, is it possible that pollination is much easier if there is more oil that the plant can produce? So successive generations start producing more and more oil in order to get pollinated.
No, because pollination is all artificial here.
Oh,
right? Because you basically just kill the bottom 90%, right? And then let the top 10% mate. Okay, I guess we stop here, but I do want to know if everybody, show of hands, if you did not understand what the fuss is all about. I mean, you could, okay.
I mean, I understood what the idea you're proposing, but I'm
नहीं,
not.
अभी तो propose किया ही नहीं मैंने, हाँ.
हाँ, नहीं मतलब समझ गया मैं that if mutation is there should be step increase, but why only step increase, that is not clear.
Cool, that's fine. I mean, that's in the 2 slides that I have to follow, which जो हम next time करेंगे अभी. But I want you guys to understand, appreciate छोड़ो, appreciate तो ठीक है, you might think, what is the big deal, it's just gone. But I do want you guys to understand why this is a problem that we have to explain. Okay, we will start with this on Friday and close this discussion, hopefully. But if you, yes.
Can I say something?
Yeah, of course.
Could it be that?
Just 1 second. I know some of you have class at 11. Please feel free to leave if you have to leave. Don't feel obliged to stay. Those of you, this is being recorded, but I will not take this discussion too far forward without everybody. All right. Yeah, Anirudh, go on.
Could it be that the higher level of oil, for example, is a dominant trait? Now, I don't know whether it's genetically dominant or phenotypically dominant, but if it were dominant, then in that case, you could expect to see a trend, right? Based on Mendelian genetics, for example, if it were genetically dominant, then you would expect to see a maximum at that particular time.
At what particular time?
At each time. So if I have a mean, which suggests that, so I've selected a mean in which, for example, 10%, in which the oil content is higher than the previous generation. And Mendelian genetics would dictate that there is a higher chance or higher population in that particular.
But let me understand this. So what you're saying is that there are so many alleles in the population, and I don't understand. So
No, I'm just saying that that each of those mutations are dominant from the previous ones for some reason.
Agreed. Sure. So, but then we are still left. But it is still left to us to explain that these mutations are increasing with such frequency.
Is there any artificially I mean, are they artificially producing mutations? It's just a thought.
No. It's just a field experiment. Nothing.
Okay. Yeah.
Okay.
Yeah.
Yeah. So exactly. I mean, we seem to be stuck here, but good. I want us to be stuck at this point, right? Because this, we will see on Friday, underlines why evolution in prokaryotes, bacteria, yeast, kind of stuff that I do in my lab, is fundamentally different from the forces that shape evolution in sexually reproducing organisms. And this is a good example. So I will just try to make sure that everybody's on the same page as far as this conundrum is concerned on Friday when we meet, and then we'll take it from there. And with that, we'll yes, that's right. And then we'll start with section 2 of the course, probably midway through Friday's class. Okay.
So,
Yes.
just out of curiosity, what was the objective of that experiment? I mean, why the oil? Did that oil had any significance in nutrient quality or something?
Yeah, corn oil, right? Edible purposes.
Okay.
So Illinois, the university, this is where I went to grad school. It's about 200 kilometers from Chicago. And for as far as you can see, and I mean, literally hundreds of kilometers, it's surrounded by corn. Okay. So there is a financial incentive in being able to produce the kind of crop varieties that would yield more oil.
Got it.
I mean, nobody does that kind of research anymore. The corn experiment only goes on because of ceremonial purposes, because it's the longest running evolution experiment in the world. So it's primarily ceremonial, but it's cool while it's still there. We'll discuss another evolution experiment with bacteria, which has processed the maximum number of generations. But this is by far, but that one was started in the 1980s. But because it's bacteria, generation time is much faster. You can process 10 every day. Corn, of course, is 1 a year. So it's not the longest experiment in context of number of generations you've processed, but it's the longest in terms of just the sheer amount of time that's gone into it. Okay.
Yes, sir.
All right.
So, the external factors seen during the experiment?
I'm sorry, say that again.
Were the external factors same during the whole experiment? Like they might have to use the fertilizers.
Yeah, I am not sure that what kind of control. Again, I am not. I'm sure I could look it up, but I haven't. I don't know of the. I don't know of the extent to which these people have gone to make sure that those environmental factors have remained the same. I'm guessing they have not. It's been a long experiment. But the issue is that but the issue is that, I mean, now we know why it's happening, and it's all genetics. Okay. It's not environment. We know the molecular determinants which have enabled this change in the phenotype in terms of oil content. But I am not aware of the controls that they have done to control for constancy of soil quality, air quality, temperature. Definitely not air or temperature. Soil, I don't know if they've done anything. But that's not it. I mean, that will of course introduce some variation, but that's not what it accounts for, what we're going to try and explain next time. But a good point there.
Okay, thanks.
Okay, let's close for today. And the new people, Gaurav, Isha, you can just stick around and I'll have a word with you guys.
Thank you, sir. Bye.
Bye.
Thank you, sir.
Bye-bye. So, Gaurav, I've added you on AC. This is the link that I sent you this morning for every week. And assignment 1 has already happened, but I will let you submit assignment 1. Today's Wednesday, maybe let's say by next Wednesday again. Okay. Is it okay?
Yes, sir.
नहीं, गौरव उसको तो ग्रेड मिला ना.
Okay, अच्छा.
गौरव सो गया शायद. All right. Okay, it's fine. Okay, cool. ठीक है. Gaurav, you can leave too. So next Wednesday, assignment 1 is due, and the details are there in the video lectures. So Isha, Gaurav, ठीक है, you can leave. Okay, Isha, tell me, what is your interest in doing this?
So
Do you
sir,
mind switching
actually,
on the video? It's just weird talking to a computer on
Yes, I can. Actually, I've connected to another class via laptop. So just a second, I can
No,
switch
no,
on
no,
my
that's
video.
okay. That's okay. I don't want to interrupt your class. We'll chat another time. You go ahead with your class.
I can, like, some other time I'll switch it on. So basically, actually, we had a course of biology,
Mm-hmm.
like in my first semester, which was the basics of what you're teaching right now.
Mm-hmm.
So I found it quite interesting. So now the plan is, if I want to go in the field of being an astronaut, so I'll have to do research. And right now I'm in chemical, so I'm doing some research part based on uh, thermodynamics or evolution of life, because in future they'll be finding life on other planets.
Hmm.
So I wanted to just get a basic idea of how things work, and
Mm-hmm.
therefore I thought maybe I'll just uh, drop a mail to you and ask if I can learn something.
Yeah, sure. So, I mean, yeah, no. So, I mean, if astrobiology is your interest,
Yes,
that's
actually.
yeah, that's fine. And there's a lot of astrobiology work going on around the world. So, I would say, uh, yeah, you don't need to work with me or anything, but you just be part of this course. You will become aware of all the fundamentals of evolution, and I
Mm-hmm.
think that can be a starting point, you know, of your learning towards astrobiology.
Yes, sir.
Yeah.
Even I was hoping the same.
Yeah, yeah, no, that's fine. No problem. But I do expect that you attend every lecture. I mean, I have two other people who are also interested, who are also attending, but are not students of IIT Bombay. And I am happy to allow people like you to sit through the lectures, uh, provided one you attend every lecture, which you can log in and sleep. You know, I can't check. Uh,
No, sir.
and 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 also submit all assignments. So as long as you fulfill these two things, I'm happy to have you in the class.
Sure, sir. I will try my 100%. Maybe if there is some exam or something, I'll inform you prior that for this
Yes,
week you can
that
excuse me.
yeah,
Hmm.
that's fine. I can. I mean, I'm happy to accommodate that. No problem.
Otherwise, I am ready to do all the like things which you asked me to do.
Sure. Yeah. No, my assignments are usually I give plenty of time. So it's not कि आज दिया है तो Friday को due होगा. I usually give at least 10 days. So.
Oh, okay, okay, sir. Good then.
All right, ठीक है. I will see
Okay,
you on
thank you
Friday
so much.
class. Bye.
Yeah, sure, sir. Thank you. Bye.